about components similar to all living organisms. Whether you're talking about a human, yourself, or an animal living in your backyard, or a little coral living in the ocean. Okay, we've talked about some properties. And then we left off with this concept or this theory of evolution. And I identified that we're just going to mention it today, and we'll talk about it in much better detail when we get into the fourth unit of the semester. We'll look at why evolution is an accepted theory out there. And then I found this image just to kind of give you a visual. Because a lot of times when we hear the word evolution, we're ingrained to think that our cousin is a monkey and all kinds of funny things. So this little snapshot of some different sea animals, right, some snails, just illustrates some of the variation of different species. Okay, and so not all organisms of the same type are identical because they have been best fit to live in their specific environments. They take on different characteristics, and then thusly they become unique species over a long period of time. And so here, just looking at a single type of organism, a snail, okay, we can see a lot of color variation because these animals would live in different environments and they need different traits to better survive. When we do look at the concept of evolution, an important thing to understand is that it is a scientific theory, and that is also a term that we will better define today, what a scientific theory is. So it's a scientific theory. And the common kind of thread behind evolution is noting a gradual genetic change over a period of time. And I don't know if you can notice this or not, but I've got the word time kind of italicized. That time span is variable depending on the species at which you're studying. A recent article that I read the headlines for talked about some whale that lives in the sea. It hasn't seen any genetic change in over 400,000, probably more than 1,000, right? A long time, right? 400,000 years, right? They've been consistent. But other organisms, like bacteria, we can see genetic changes within a few years. We can observe these evolutionary changes. But it's a gradual change. You're not going to see profound, strange mutations in species where they look like one thing one day and then completely something different the other day. It's a gradual, modified change over time. And the whole point is to give diversity to the environment. Okay? Evolution provides a source of diversity, biological, genetic diversity. Diversity is good, right? The evolution is a scientific theory. It's just a genetic change over a period of time, and it gives diversity in nature. It's capable, possible, through species' ability to adapt, a process called adaptation, to adapt to their environment. Species are able to adapt to their environment and survive. And then those animals, or organisms that are able to adapt to their environment, not only survive, they do what? Multiply. They what? Thrive. They survive, they thrive, and they multiply, or they reproduce. Okay? And that ties into this concept called natural selection. Okay? So, how can we define, I kind of defined natural selection. How could we better define natural selection? Survival of the fittest. And not just survival and, we want to survive and reproduce. Those organisms that are best adapted for an environment survive and reproduce. And when they reproduce, there are more of their genes out in the environment. Okay? And so another addition that you can talk about in your natural selection notes is They just have a greater proportion of their genes in the population. Genes. Genes. There's a greater proportion of their genes in the population. Dependent upon this concept of natural selection. It's not the only thing, but it's an important driver for evolution. We'll give some additional detail when we get there at the end of the semester. But during natural selection, those organisms that are naturally selected, they are better adapted to their environment. Okay, so they can survive, and despite what conditions may be changing, they're able to survive in those changing conditions. So are we topically kind of okay with evolution, a genetic change over a period of time, because certain species are naturally selected because they can adapt to their environment? All right. Those are some important components of... I just talked about this. We talked about all of these components. We said natural selection, an organism survives, it also reproduces, it gets its genes in the environment, and they have a greater percentage of their genes in the population. So I kind of walked us through this, started off showing that along with the discussion. Good to move on? All right, and another visual analogy looking at natural selection, we have some beetles, some different color patterns. Okay, obviously in a dry environment, between the green and the brown beetle, which one's going to be better suited to survive? The brown. Okay. So genetically speaking, it's better adapted, it's going to survive, reproduce, and adapted to their environment, so it's better survive. Also talked about adaptation. Once an organism adapts to the environment, it's going to be some type of trait or physical characteristic that is eventually going to be passed on to the offspring. It allows for the offspring to also be better suited to that respective environment. And that contributes to the reproductive success that I mentioned earlier. I always played some tie in with that. These things, natural selection, evolution, and the other properties of life that we talked about, there's be some commonalities of different organisms. Whether they're, as I mentioned, something living in the sea, or maybe a bacteria living on your skin. I have this image here as a prompt to illustrate for you that we can certainly see evolution in our society today. And I believe we talked about it when we started class a week ago. We talked about that MRSA. Did we talk about the methicillin-resistant Staphylococcus aureus? You've probably heard of it. You've heard of a staph infection, or you've heard of MRSA. Those bacteria have become adapted to their environment. What are we putting in the environment that they can adapt to and survive in? Well, the bacteria we might call a germ. Okay. What do we take all the time? I feel a little sick. I better go to the doctor and get what? Antibiotics. Okay. That would be an example of something that bacteria can adapt to and survive in. And so we take antibacterials all the time, even when we don't need them. We put them on our hands all the time because we think it'll keep us healthy. But we're challenging the bacterial species. They're adapting, they're surviving, and they are reproducing. Now their genetic changes are out in the population. We give them a drug that they can now survive. What happens? We try to get a stronger drug that may or may not work, right? And we just keep pushing this forward. So you can see evolution very much in our society. You just look at some bacteria that are surviving these antibiotics. They are evolving. They're adapting to the environment that we're pushing on them. So it's going to be interesting how we as humans are going to survive something simple like the bacterial infection of years when basic penicillin or something isn't going to phase it. Okay? So what we talked about living organisms is we said they're organized, we went through the hierarchy. We talked about they're going to have some form of energy conversion. We talked about the three primary categories. We said cells or organisms must reproduce and grow. We also talked about the ability of cells or organisms to be able to evolve in order to adapt to a changing world. What we added to the environment or to the list is that something living should be comprised of at least a cell and have DNA in it. And we also added to the list on Thursday that our cells should also be responsive in the environment. Okay, so that's going to summarize our section 1.1 discussion and get us into section 1.2, which is going to look at this concept called taxonomy. Okay, so taxonomy, does anybody know what it is? Excellent. The naming and classification of organisms. Taxonomy is the naming and organization of living organisms. It's a biological classification system. It'd be analogous to how a library is organized into sections and you find individual books, but taxonomy is utilized to organize living organisms. We'll study it at a pretty broad category. We'll talk about the primary kingdoms, and first even domains and kingdoms. We'll discuss those today. And then we'll kind of leave the intermediate structures. You probably remember these maybe from public school. If you went there, phylum, class, order, family, etc. And then we'll kind of hop to the end and talk about what a genus, and then more specifically a species is. So it's just a classification system to find some commonalities between different organisms to make it a little bit easier to understand. And then just a visual analogy for you that maybe you can appreciate better is just how you might order files in your computer system, right? So you got your C drive, and then from there you got your personal files, and then there you've got your, you can keep going on. So it's just a classification system to understand diversity here on Earth as we know it. It's just a classification system. And as I also identified, it gives a hierarchy to our organization. We'll identify the three domains in just a moment. We're going to talk about eukaryotes and bacteria and archaea, and then we'll just further discuss the kingdom subcategories that we'll find in our eukaryotic organisms. We'll pretty well for this semester focus on these things called eukaryotes. There are three domains of life if we utilize this taxonomical hierarchy. The three domains include domain archaea, as I mentioned, domain bacteria, and domain eukarya. You'll notice I just have the letter D to represent domain. Okay. So let's talk about some characteristics of each of these. Common characteristics between these domains is that they are all going to be comprised of at least one cell. Okay, so each of these will be comprised of at least a single cell. And maybe I should better clarify this. Domain archaea and domain bacteria exist as single-celled organisms. So the first two domains, archaea and bacteria, exist as single-celled organisms. and the bacteria, they exist in our environment as a single cell. The entire organism is just a little, tiny little microscopic cell. Conversely, domain eukarya, or the eukaryotes, may be single cell, or they may be multicellular like yourself. You're a multicellular eukaryotic organism. But not everything, not all eukaryotics are multicellular. There are some single-celled eukaryotic organisms. A note about the archaea is that they live in extreme environments. The domain archaea are organisms that live in extreme environments. Can somebody give an example by chance? Like the tundra, so extreme cold or even extreme heat. Even more hot than that, like our subthermal vents that we have, like just even more hot than the desert. Okay, good. What else can you think of besides temperature that might be hostile to something else that's not an archaea? Okay, good. So lack of oxygen. There are environments on Earth that there is no oxygen, and so some archaea have the capacity to live there. Good. Not only, say, just oxygen, but other chemicals, sulfurous environments, really acidic environments, environments that would kill something that's not an archaea. The archaea are adapted to survive and thrive in. Okay, so they're pretty extreme. They're sometimes called extremophiles. They live in extreme environments. So beyond just introducing them, we won't go into much detail on the extremophile. Conversely, the second domain that we'll study are called bacteria. You've heard of those. We just gave an example of some of those. They are certainly single-celled organisms, and they are considered to be prokaryotes. Bacteria and the archaea are categorically called prokaryotes. P-R-O-K-A-R-Y-O-T-E. So you might know what that means? A prokaryote? They have no nucleus. No nucleus. We'll identify what that is in a moment. Our bacteria and our archaea are prokaryotic, which means they have no nucleus nor any other membrane-bound organelles. And this is common to both bacteria and archaea. Not only do they not have a nucleus, they have no other membrane-bound organelles. Correct. All archaea and all bacteria, correct. Lack a structure called a nucleus, as well as they also lack any other membrane-bound organelles. Which may not mean anything to you right now, but hopefully it will soon. The eukaryotic organisms, the terminology describes them, and so they must have, right, by process of elimination, I said what prokaryotes don't have, so eukaryotes must have a nucleus and membrane-bound organelles. Our eukaryotic organisms do have a membrane-bound nucleus and other organelles inside of them. We also identified that they may exist as multicellular organisms. are some of the just general kind of highlights of those types of organisms. Any questions about those major kind of differences before we move on? Yes.
Say that the eukarya can be single-celled and multi-celled?
Yes. Yeah, or. So if something is designed to be multicellular, like yourself, you would always be multicellular after fertilization. However, there are some single-celled organisms that are eukaryotic that can, say, live in, say, pond water. They are eukaryotic, and they're always just single-celled. So you're correct, but I just changed your and to or. We don't flip-flop. Good. Additional questions? I want to go ahead and elaborate a little more on the different domains, the kingdoms, really, of the eukaryotic domain. There are 4 kingdoms of our eukaryotic organisms. The first is called, and again I've got the abbreviation, the K for kingdom. The first is called fungi. You've probably heard of a fungus before. The fungi kingdom, categorically, remember we talked about the different types of feeding, energy consumers, sources, etc. The fungi are what's called consumers. Okay? So they can get their nutrients from the environment. Our animalia, or our animals, are also consumers. They have to eat other organisms. And our plant kingdom, they are categorically autotrophs. That means what? They can produce their own food by utilizing sunlight. And then the 4th category is called kingdom protista, or our protists. And these are kind of you have that stereotypical kitchen drawer where you need it, you don't know where it goes, you just chunk it in there, right? Okay. The protist is the kingdom for our eukaryotic organisms. That is the kitchen drawer. Okay? If there's an organism that is not clearly a fungus, or an organism that is not clearly an animal, nor a plant, well, back when this system started, they said, well, let's just call it a protist. And we just throw it in there. And so we'll see a mixture of different type of feeding sources. And so the protists, some of them are autotrophic, so some of them have the ability to photosynthesize. Some of them have the ability to go out and consume nutrients. Some of them are motile, some of them are not, right? So it's just kind of a mixed bag of different types of organisms. And each of these kingdoms you will spend a week on in lab. So you'll have a little bit of additional detail on the different types of eukaryotic kingdoms when you get in there in the class. Okay? Fungi, what are some examples? What do you think might fall into the fungi kingdom? Mushrooms. Mushrooms, a great example. Animals, for me, I just cannot retrain my brain. Now, I always think of something like a vertebrate, a dog, an animal, something, right? But even the example I've given earlier, like corals that live in the ocean, those fall into this category, but they're invertebrates. Okay, so if you think about like an earthworm, those all fall into the category of animals. Yes. They're called chordates, phylum chordata. Yes. That would be a plant. It's actually a plant. It has leaves and vasculature. It's characteristically a plant. It just has a very interesting way to trap. I don't know if they do eat the flies. They do get nutrients, but they also have the ability to photosynthesize. I'm not sure about. There's always weird examples, right? Venus flytrap. I'll have to look up why they do that. There's some benefit to eating or I guess killing a fly. I don't think it's for pollination. I'm sure they must probably extrapolate some nutrients from the fly, but they are a plant, and they have the ability to photosynthesize as well. And they have showy flowers. Cool. Good question. Right, so our eukaryotic kingdoms: fungi, animals, plants, protists. I was supposed to hide that other slide. I was trying to make it look pretty. But anyway, so we talked about the fungi. To be more accurate, you can clarify that, and really they're more decomposers than consumers. I had a term up there. Okay. Just like we showed a relationship before, it's the same table, so just looking differently. But we can see a relationship of how fungi provide a source of nutrients, say, for plants, and plants provide a source of nutrients for animals, okay, and the protists kind of mix in there, and the bacteria. All of these organisms are very much interdependent upon one another. So those are our four kingdoms. If we look at the relationship, if we step back and look at all of the domains, we said our archaea bacteria are called prokaryotes, and everything else is a eukaryotic organism. If you look at this image, though, it's there to illustrate that the bulk of living organisms on Earth, are prokaryotic. We can't even see them without the naked eye, right? So there's organisms living everywhere that you may even acknowledge that live there. So we are able to see the multicellular organisms, okay? Or we could presume sometimes bacteria when they're present, when we get sick, or we need medicine, something like that. But there's a lot of diversity that you can't even see without using a microscope to acknowledge that they exist. When we look at talking about the different species, it's important to understand the nomenclature utilizing this taxonomy. And so one thing you'll practice in lab and get in the practice of writing in class is how to properly write the genus and the species. And so when you do this, go ahead and write these rules down. The genus is always capitalized. The species is lowercase. And when you're writing it, if you're handwriting it, they need to be underlined. Or if you're typing it, you should italicize it. So again, the genus, the first name is always what? Capitalized. The species always lowercase. If you're handwriting it, you should underline each. Or if you're typing it, you should italicize or underline. Additionally, what can you do with the genus? Just abbreviate it. Okay, so you can still have the capital, and you just abbreviate it, and then you still spell out the entire species name. You've probably heard of E. coli. The E is the abbreviation for that bacterial's what? Genus. Anybody know what it is? Escherichia coli. It's easier to say E. coli. Some other examples include, right, you call the alligator. Well, its genus is appropriately called alligator. Okay, so that one's easy. I'm not even going to try to mispronounce that, but that one must be from Mississippi, right? I was trying to make it sound fancy. Like a house spider, okay, genus and species. Here's the fly trap you asked about. I actually have an image of it. Dionea muscipula. Okay, so I know I'm butchering that, but to write it though, you can kind of see some commonalities on how to capitalize the genus and lowercase the species. I found these images from the National Wildlife Foundation. Foundation. You might want to go there, some really cool diversity images, and you might want to go ahead and look up about that Venus flytrap and answer your question. So why does it do that? Eat that poor little fly. So National Wildlife Foundation, nwf.org. I'll show you some different types of examples. Any questions on how to write the genus and the species for a particular organism? I can guarantee in lab you'll have a quiz over that, so get in the practice of writing that properly. All right, that transitions us now to the final part of the chapter, just talking about science, and that's what you'll study in lab this week, looking at the scientific method. And scientific method is another one of those things that everybody's got their opinion on. Is it good or bad or whatever the case may be? It's fairly well accepted by a lot of people, so we'll go ahead and present science and biology to you utilizing the scientific method because it works pretty good. So just like everything else, of course, some things are fallible or not accurate for everything that you might be studying. And if we look at this thing called the scientific method, and before you even look at the steps, and before you start writing everything down, what do you think that the scientific method is, or why do we have it? Very good. A general way to answer questions and do what? Test ideas. So a way to find answers, right? Curious about something? Well, let's design an experiment that will kind of help answer our question. We'll do it in a logical manner. Okay, so it's just a tool, if you will, to answer questions. But it's designed so that there aren't any errors, if you will. So we don't have any too many mistakes, right? So it's more controlled. So we're going to go through the kind of published steps of the scientific methods. The first step is to do what? You've got to make an observation. You've got to be coherent. You've got to pay attention, right, to the environment. You can make an observation directly. You can go out there, and I can walk out this morning as it snowed overnight. Okay, I made that observation. I didn't have to wait for the news to tell me or whatever. I saw the snow in my car. Okay, so you can actually observe, like physically observe something. However, maybe you could read a piece of literature written by your favorite author, and you make an observation. Okay, so it could be something historical, a piece of literature that you've read. Okay, you don't have to physically see everything. Maybe you made an observation based on a previous experiment that you've done, or another something else that's happened to you in the past. Right? You just note something. You make an observation, right? Something piques your interest. After you make an observation, what should you do then? Curious mind would then pose a question. Okay? The second step to the scientific method is to asking a question. And the question should be very specific. The question has to be very specific. If it's not specific enough, then the subsequent steps may not give you the proper answer. Okay, so it has to be a very specific question. Then we would form a what? Hypothesis. Okay. What they tell you in lab? Anybody go yesterday? Read ahead. What do you? Yes. Do what? Okay. So, hypothesis. What do you think based on your reading? What's a hypothesis? Very good. It's a testable explanation for your question based on your observation. Okay? So, key thing is that it is very much testable. Okay? A hypothesis is a testable statement. Additionally, it's very what's called tentative. What does that mean? It's not proven, and it's subject to change. Okay, it's a tentative kind of explanation that we can test. Because it's tentative and it's testable, it might be proven what? Wrong. Okay? This right here, this third point that I have listed, is very difficult for a lot of people to acknowledge. Okay? And this is where we see sometimes bad science coming in, okay, to play. A scientist may spend decades working on something and they think it's X and it turns out to be Y, and they're like, well, don't care, I'm publishing X, right? And those are the people we see getting hauled off to jail and all kinds of good stuff, or if they're in another country, worse than jail. So it's important to understand when you're designing an experiment that you may not be right. Okay? If you've lived, you know that you're not right 100% of the time. If you are, you might want to rethink your perception of yourself. Okay? You're probably not right 100% of the time. Okay? And so the hypothesis, when you take lab, you're going to design it as an if-then statement. Okay? So, for example, if I wanted to test if it snowed overnight, okay, I could have designed a test before I walked out of the house. I could say, if it snowed overnight, then there will be snow on my car. Okay. I could test that, right? Okay. I could open the door, take my head out, and there's snow in my car. Okay. In that case, I don't have to do much experimentation besides open the door. Okay. However, I could have stuck my head out, and there was no snow in the door, right? And I could make my conclusions. Okay. So where I was going is your statements in class in your lab will be that very simple if-then. Okay. Obviously, in science, they'll be a little bit more developed, okay, than just a simple if-then, but we'll kind of keep it simple for a basic appreciation. So a hypothesis is certainly a tentative explanation. We're going to design an experiment, and it can certainly be proven wrong. We hope we're right, but we may be wrong. What's the next step then? I kind of lumped it in that explanation. You've got to test it. And so you've got to design an experiment. This is the difficult part to making sure the whole scientific method is accurate. You've got to design an experiment. And the experiment has to be well controlled. And we'll identify some of the controlled factors. It also has to be capable of being replicated. So you've got to design it well enough that you could redo it, and your notes are good enough that someone else could redo it. Someone in another country could redo it with the same materials and procedures. It's got to be reproducible. If it's not going to be reliable and just not reliable. It's not going to work. Okay? So we'll come back to what should we control in the experiment. After the experiment is controlled and conducted, what should we do? Collect the data. Okay? Data. Science is all about data, however you want to pronounce it. We've got to collect the data, but most importantly, you need to analyze it. Okay? You need to interpret your results. Collect the data and analyze it. Once we collect the data, we analyze it, what can happen? You draw your conclusions. You interpret that data, you draw your conclusions, and the big thing is you either accept or reject your what? Your hypothesis. So I was either right or I was wrong. On your notes, I want you to make a modification, or maybe where it says an additional comments. I want you to acknowledge when you interpret your data, you're making new what? Make a new what? Making new observations, and I might be forming new questions and forming new hypotheses. So, I want you to acknowledge that science is a circular, never-ending process. Hopefully you never stop making observations and being curious about the world around you. So on your notes, you can make a loop back from 6 to 1, or just in your comments say, well, once the data is analyzed and interpreted, you're probably going to make new observations and design a new experiment. What questions do you have about just the kind of general major steps of the scientific method?
It says in the book that you don't always need an experiment to do it based on investigational observation.
Yes, like me poking my head out of the door. I don't need to calculate the number of steps it'll take me to get to the door. I don't need to make sure I open the door 10 times. Yes. But if I want to investigate a drug on cancerous tumors, I better design a really bulletproof experiment. Okay, so it depends on the magnitude of what you're studying. That's correct. Additional questions? All right, so let's look a little bit at the design of an experiment. And maybe I have, turning to the page 6 of your handout from chapter 1 if you're using it. An experiment. Sample size is critical. What does this mean? What does that mean? It would be a factor that is controlled, but what does that mean, the sample size? The number or amount of whatever it is you're studying. Okay? So for example, if I'm doing a human drug test, I want to test not just the effect of the drug in one person, I probably want to test it in hundreds of people before I put it out in the market to be exposed to millions of people. Okay? So the sample size has to be adequate enough so that it represents what you might see out in nature. Going into that, though, is going to be cost. Obviously, the more numbers you have, the bigger the sample size, the more reliable the data is. But at some point, the data is going to be, there's going to be too much. So you don't want to have too many samples, but you certainly want to have enough, right? So the sample size number will depend on what you're specifically studying. Okay, so 10, 20, depends on what you're studying, maybe hundreds, right, of samples that you're measuring. Another thing that we need to take into consideration are our different variables. The first is called the independent variable. Independent variable. We're not looking at the word independent. What do you think the word variable means? A level of something that could be, you're right, but you're getting really specific on me. Just the word variable. If something varies, it changes, right? Fluctuates, okay? And so we're looking at factors that could change an experiment, okay? So if we have a variable, we have an independent variable. Now let's give your definition of the independent variable. Restate the independent. Okay, so the independent variable of an experiment is that which is, what word did he use? Manipulated or changed. Okay? So it's really what you're investigating. The independent variable is what is changed in the experiment. So if we keep some things changed, we probably should keep some factors consistent so we don't have too much variation going on. So we have an independent variable. What is changed, what is manipulated. There must then be a dependent variable. And the dependent variable is actually what the scientists would measure, where the data is actually coming from. And we'll give some examples in just a moment. So the independent variable is what's being changed. The dependent variable is what is being measured. There's also something called a standardized variable. And these will be factors that are constants in the experiment, so that they don't influence the what? So we don't influence falsely and what part of the experiment? The outcome, which would be your what you're going to measure, your dependent variable. So standardized variables are factors in the experiment that are held constant between samples, not to falsely influence the output. I'll come back with some examples. Okay. Let's just give the definition of the rest of the terms. Also in an experiment, we should have a control group. What should a control group not receive if we want to see if what we're testing does or does not have an effect? Potential, we would call it maybe a placebo. In those list of 3 things we had earlier, the should not receive the independent variable. The control group is going to be a test population does not receive any independent variable or no treatment. Independent variable receives no treatment. Or independent variable. Control group receives no independent variable. So that is the yes, kind of. Placebo is a term utilized for human studies, and so not everybody just studies humans. So it would be like the placebo effect, though. It'd be like taking a pill, but there's no medicine in a pill. So yes, it would be analogous to a placebo. And the last item that I have on your list is statistical analysis. You might know about this. Obviously, I want to show what is statistical analysis. Analyzing the numerical data. Okay. Well, once data is collected, the data needs to be analyzed statistically. So superficially, something may look like it may have an effect, but once you put the statistics to it, it may not be that big of an effect. Okay, so it's just a mathematical interpretation of the results. Let's go back and answer some of the questions that you guys had that just put into practice. So for example, okay, let's say someone wants to study the rate of oxygen production, okay, for yeast cells growing with and without cyanide. Okay, and cyanide is known as a respiratory inhibitor. What would be the dependent variable? And remember what was our dependent variable? That was the what is being measured. So what are we measuring in that experiment? The rate of oxygen production, or production, in that scenario. We're measuring oxygen production. What would be the independent variable? What would be the treatment? The cyanide would have been the treatment, or the what? Independent variable. What would be a good standardized variable? Yeast cells, what? Remember, our standardized variable receives any, which is in this scenario, no cyanide. Okay. So in this case, our standardized variable would be yeast cells without our independent variable, without our drug, right, the cyanide. Lori, what question do you have? Okay, so let me go back. Let's see another example, okay, that I have in here. So someone wants to study the number of colorectal tumors for someone taking aspirin once, twice, or 3 times a day. Okay, so that's our experiment design. To go back our definition, our independent variable, this generic independent variable, how do we define that? Okay, what you're investigating. The dependent variable is what's being measured. The standardized variable would be what's held constant and not without any treatment, right? Independent variable. Okay. So in this case, what would be the independent variable? The amount of aspirin. The amount of aspirin. It would be the independent variable. What about the dependent variable? The number of tumors is what we're measuring. Okay. Does that help a little bit, Lori? Yes. Additional questions? I'm kind of like running you through this because you guys will spend an entire hour practicing this in lab this week. Okay, I just want to kind of address the terms today in lecture. All right. Not overlooking something. There's a little section in your handout about additional questions. Okay, the question is an aspect is obviously, well, what about things that are a lot more difficult to answer in society? Like, is there any type of taxonomical relationship between, say, birds and dinosaurs? Well, clearly I cannot I can't go out and see a dinosaur. Okay? However, I can go out and collect fossil evidence. And now we have DNA evidence. Okay, so some of the DNA is preserved in some of the fossils. So there are tools that are available out there. So you might need something besides just going out and physically doing your experiment. You might need historical observations, genetic tools. Right? You might have to open up your toolbox a little bit more wide than just peeking your head out the door to see if it snowed. Okay? And so we can address difficult questions. It's just going to take a lot more investigation. And that's the problem with these is it's open to interpretation, analysis. And so that's where we get these opposing views of these really kind of big questions that we have about heredity and etc. Okay? A couple of just notes. This statement, right, nothing is ever proven. It's difficult to accept, but I think it's a pretty good accurate description of science that we can. Nothing is ever proven because science is always what? Changing. Our knowledge base is always changing, and everything we know is considered to be falsifiable. For example, you remember the planet that used to be, or the, I don't know, was a super dwarf, whatever. Pluto, you remember Pluto? It used to be one of our planets, and now it's like squashed down. It's no longer a planet, right? So we used to think it was a planet, now it's not technically a planet. So our knowledge base is always changing. We're always learning. And so what we know today may not be 100% accurate, say, in 20 years, 50 years, 1,000 years from now. Okay, so it just depends on the new data that's coming in. And with our technology that I identified the other day, our data is coming in at just an exponential rate. We're just gaining so much data. We're learning a lot about cells and all types of different types of organisms. And if we look at the relationship between scientific knowledge, there are 3 main levels of explanation. If you remember, we talked about the hypothetical level, the hypothesis, okay? you're going to form some hypotheses today. Remember we said these are very tentative and falsifiable. And they're not solid, right? A single person can form a hypothesis. They're right or they're wrong at the very end. However, we know that there are more well-known facts out there. And the next big leap to scientific knowledge is called a scientific theory. Scientific theories are based upon multiple what? And hypotheses that have been proven. What? Correct. In a theory, there is, and I'm going to use a pretty ambiguous word, lots of data available to support scientific theories. Many people, many researchers, not just one person can develop a theory, but many researchers' combined work can help form different scientific theories. And I'll give some examples in just a moment. The next level is a scientific law. What do you think about a scientific law then? They're more than a theory. They have been proven correct. I hate using that word proven, but I'm going to use it. They're well accepted. And another note that you can identify with a scientific law is they are generally associated with a mathematical formula. So something that you can apply. You could sit there and calculate out. So your laws are usually associated with a mathematical formula. I'll list some examples. You guys will come up with your own hypotheses. Anybody can come up with a hypothesis on their own. Some examples of theories that you may be familiar with, or you will become familiar with, for example, will be the cell theory. We'll talk about it in chapter 3. You may be a fan of the theory of gravity. I am. I believe it. Can't see it, but I believe in the theory of gravity. You may have heard of the Big Bang theory, theory of relativity, theory of evolution. There's a whole bunch of evidence to support these cell theories, but there's no solid, definitive, worldwide accepted view mathematical application. So our theories could still be what? Disagreed with and falsified. But there's plenty of evidence out there to actually support them, and that's why they're out there and widely accepted. Some examples of scientific laws, the way our planets orbit, okay, mathematical application to those. Newton's laws of motion. There's a commercial out there, an old man sitting on the chair, not old man, but an aged man sitting on the swing, right? He's like, object in motion stays, right? Or whatever, at rest, stays at rest, object in motion stays, right? Those types of laws of motions. Our laws of thermodynamics, you'll cover, I believe, in chapter 4, maybe chapter 3, I can't remember exactly. Those are some examples of our scientific laws that have some mathematical application to them. So, that fairly well concludes our chapter 1 discussion. And I have 2 minutes left, so I'm going to use it. I'd like to go ahead and get into chapter 2 because it is a pretty heavy hitter. And as I noted earlier, I made the file but didn't give it to you, so I'm going to go ahead and hand you the first 4 pages, but it's a 10-page document. So if you have what I've given you, you'll need to print page 5 through 10. To address chapter 2, which I mentioned there's a lot of information in the second chapter. And in theory, it probably could have been easily broken up into 3 chapters. and the chemistry, and then the macromolecules, but we'll go ahead and address it as a single chapter. And the objectives that we're going to study, I did go ahead and put those on your handout for you today, unlike what I did for chapter 1. But we're going to talk about basic atoms, basic chemistry, basic bonding, and then the different macromolecules that are important for biology. We're going to talk about pH, water properties, and then we'll conclude the chapter with how these different macromolecules are actually formed, because for the rest of the semester we're going to be putting into practice how these molecules work. So although a little bit scary, this chapter 2 can be, I'm going to encourage you to really go back, repeat it, address it regularly, frequently, because if you don't, it's going to be really scary. Okay, so I want you to become more familiar with it. And what we're going to do is we're going to start out with the levels of organization. And then we get a chance to kind of fantasify this with the images, so hopefully you kind of get along. But a term that you've probably heard before and maybe utilized before is called matter. And matter is anything that takes up space and has a mass, a measurable mass. Matter is anything that takes up space and has mass. So the table that you're sitting at, the pen you're using, has mass. We are going to focus on what's called biomass. What does that mean? Living things, right? The mass of the living things on Earth. Okay, biomass. But just in general, mass is anything that takes up space, or matter is anything that takes up space and has a mass. Weight is sometimes another word that you could use, but it's not an accurate description. I can appreciate that term. Alright, so the question is, well then how can we better define matter? Okay? And on Earth, matter is made up of these things called elements. You might know what an element is? Very good. It's a pure chemical substance. An element is a pure chemical substance. There's already identified. It can't be broken down to anything else, to any other substance. It's just pure. Elements are just pure substances that cannot be further broken down into something different. Okay? If you look at the elements, okay, some examples would be sodium, chlorine, calcium, the things that you'll find on the periodic charts. In biology, there are some important elements, and they're called essential. You're probably going to guess they must be danged important, so important they're essential, okay, to life. Some examples of essential elements include, you don't have to list all of these, maybe you just want to give an example of, like, you've heard of carbon. What does H stand for? Hydrogen, nitrogen, oxygen, calcium, you guys are good. Potassium, K. Does anyone know what P stands for? Phosphorus. Sodium's tricky. Na is sodium. And here, sulfur, chlorine, magnesium. These are all examples of elements that our body needs for chemical reactions or to build cells, provide nutrients, etc. And we'll find these in larger quantities. Some elements are considered to be trace elements, and we must need these in minute amounts, tiny amounts. We need our trace elements in tiny amounts, so trace amounts. Like some like iron, I don't know if that's important element. Say, for example, you've heard of hemoglobin in your what? Blood. Iron's critical for the function of those proteins. Okay, and we've got some different examples. Other examples: boron, trace elements: boron, chromium, copper, iodine, iron. I mentioned tin. Okay? You don't need to list these. I just want you to acknowledge they are needed in what? Trace amounts. Okay. And what I'd like to do is conclude today with the definition of a compound. What do you think a compound is? A mixture of different types of elements. Okay? It's a mixture of multiple elements, and we can know the ratio of their combination. Thursday. I'm going to ask you to read ahead. Use those handouts that I've given you as kind of get through the text. It follows along with the textbook. So basically read the textbook and take notes. Additional practice is coming around in this handout discussing the different types of macromolecules. This will be your first in-class homework assignment, if you will. And remember, 10% of your grade will be these handouts. So I just need you to go through, read your book, and differentiate the different types of macromolecules. Just study carbohydrates, proteins, and on the back, nucleic acids, and lipids. I want you to notice the name of the monomer subunits, maybe draw an example of what's called a polymer, and just list some additional notes about the synthesis or biological function. You'll notice that lipids are kind of unique, so complete this table. You might want to add pictures or something, but bring it for credit on Thursday. Have a good week in lab, and we'll see you on Thursday. Yes, ma'am.
So where's the book? Is it just like not the book, or is there
It is, yes, and this will be at the end of the text. End of chapter 2. It'll cover quite a bit. We'll start with
I didn't have my laptop in my bag. It's so freaking heavy.
the freaking literature book is like this big, but it's that big.
Yeah. The biology book.
Since you can